Hello, everybody. My name is Ashleigh Bope, and I'm a Ph.D. student at The Ohio State University. I'm going to talk to you today about indoor environmental dust collection techniques for accurate microbial exposure estimates. And so as a brief introduction, we spend about 90% of our time indoors. And now with COVID-19, we're probably spending a little bit more time indoors, especially in our own homes. And in these environments, we're exposed to a complex mixture of microorganisms. And this can impact human health, especially in vulnerable populations such as asthmatics or those who are immunocompromised. And so looking ahead to how climate change may impact these microbial exposures indoors, we know that extreme weather patterns caused by global climate change increases the risk of flooding events. And these flooding events can create indoor moisture problems, which then allows for increased fungal growth. And that increased growth can cause respiratory health problems for the occupants. And so when we're looking at exposure to these microorganisms, it's often happening through the resuspension of dust, especially from carpet. And these exposures can be quantified by measuring the environmental DNA from these microbes. And that's done using polymerase chain reaction or PCR technologies. And so the advent of these DNA-based measurements has really helped us to reduce the biases inherent in previous culture-based methods. But these methods are greatly impacted by sample collection methods. So we need to be aware of the collection efficiencies associated with these methods. In this work, we aim to characterize the collection efficiencies of two different carpet dust collection methods to better estimate microbial exposures indoors. And so to do that, we spiked dust with a known concentration of bacterial cells and also fungal spores, and then embedded that dust into nylon carpet. And we have two different pile heights we used. We use low pile, which is about 1 centimeter in height, and then medium pile about 2 centimeters in height. Once the dust was embedded into the carpet, we needed to vacuum that dust out in order to measure the microbial DNA present. And to do that, we used two different methods. We used a thimble filter method, which is what is depicted in the schematic at the top, and the dust is then collected inside that filter. We also have a nozzle attachment. Both connect to the vacuum in the same way, but in this nozzle attachment, we have this flat filter that it's the dust is being collected on. And so we vacuumed using these two different methods. And when we look at collection efficiencies using the thimble filter for the low pile carpet, we were able to collect about 95% of the dust. For the medium pile carpet, not as much, which seems kind of logical. We were only able to collect 56% of the dust from the carpet using the nozzle attachment. For the medium pile carpet, it was about the same. We were able to collect 54% of the dust that was embedded, but for the low pile carpet, we were only able to collect around 70%. Which is lower than what we were able to collect with the thimble filter. And so if you're tracking these efficiencies along that sample collection process, which I showed in the previous slide, when you're looking at how much DNA you're able to collect from samples, in this particular example, we were only able to collect around 19% of the DNA. From the low pile carpet, we were able to collect around 93% of the dust from the carpet. So when you combine those overall, we're only able to collect around 17% of the microbial DNA that was initially spiked into the dust and embedded into the carpet. So I really want to call your attention to this underestimation potential that is present based on these results. And so carpet collection efficiencies primarily depend on pile height types, but also the majority of DNA is lost during the DNA extraction. So when you combine these, we're looking at an underestimation potential, potentially up to 11 times more than what we were able to actually detect. And so in conclusion, incorporating these collection efficiencies is very important to avoid underestimation of microbial exposure. And that then has important implications for those with allergies and asthma. So we're only really able to measure a fraction of what is actually there. And so with that, I would like to acknowledge the Alfred P. Sloan Foundation for funding for this work, and also David Cormos for help with this project. And those are my references.